Today we're going to talk about the scientific methods. Now the reason we say methods with an S and not method is there's obviously a lot of different methods scientists use. For example, digging around in the dirt, discovering fossils. Maybe this woman is trying to insert a gene into a plant to give it its own pesticide, or maybe working with patients, giving them placebos and other patients a drug with an active ingredient we might think will produce a change, or maybe just trying to find a gene that controls something in a person. So in all these different types of scientific investigations, there are certain methods that scientists use, and again we call those the scientific method. They typically begin with some observations, some background information, maybe some research. Based on that, there's typically a question that's asked. Why do plants grow faster with certain nutrients, for an example? Based on that question, it's answered with a testable statement, what we call a hypothesis. And that hypothesis allows us to make a prediction, and we'll make a prediction using typically an if-then statement to predict what would happen in an experiment. And the hypothesis with the prediction then allows us to test it in a controlled experiment. That experiment may support the hypothesis and allow us to make other predictions, or it may not support the hypothesis, and we may have to go back and come up with alternate hypotheses. So a hypothesis, again, is basically just a testable explanation for a problem. Sometimes you've heard of a hypothesis as an educated guess. Well, it's not really guessing, because typically, again, we have some background information that we're using to make that statement. We're not just randomly guessing about it. And typically in biology class, I'll ask you to use an if, then, because statement, right? So if we add phosphate nutrient to this plant solution, then the plants will grow more rapidly because phosphate is a key nutrient for plant growth. So that would be a very good hypothesis, or a prediction that was used based on a hypothesis that we can then test, and we can test that in something called an experiment. And the experiment that we're going to do in biology class typically are what we call controlled experiments. In other words, we're testing the hypothesis by collecting information under controlled conditions. Now, what does that mean, controlled conditions? Well, there's a lot of variables that will affect, say, plant growth, and we want to keep all those variables the same, what we call constants. So in other words, if we think a certain nutrient like phosphate is going to increase plant growth, then temperature and amount of light and amount of water and all those things we want to keep the same. So the variables that are kept the same in the experiment are called constants. So that one variable that we do change is called the independent variable. So in this case, it might be the phosphate nutrient or nitrogen or some other key nutrient that we think might produce a measurable change. So that's what we call the independent variable. Okay. Now a lot of people confuse the independent variable, which is the variable that we modify and control, with the dependent variable. And you can think about the dependent variable as the change that results from the change in the independent variable. And an easy way to remember that is to think about the dependent variable depends on the independent variable. So if I give this nutrient that I think will produce an increase in growth, then I will get a measurable increase in the growth of the plant or the mass of the plant. So the dependent variable is typically the results, and this is oftentimes what we'll graph or put in our data table in our biology labs. And typically we want to be able to know if that produced a change, so we have something called the control. The control are the subjects that do not receive the independent variable. So if we have some plants that we're given a certain nutrient to, we want to have other plants that just get pure water, right? And the same amount of light and the same temperature and all those other constant variables, but the only difference being that independent variable that was given to some plants, our experimental plants, is not given to the group of plants that we call the control group. Okay? So we have to have the control to see if there's a measurable difference because of that independent variable. Okay? And these hypotheses over time, if they're supported and supported again, we can develop theories. And scientific theories are explanations that have been supported by evidence obtained from many experiments, and essentially supporting hypothesis over and over. And theories can be modified and they can be changed, but the key thing about a theory is that it must be like a hypothesis. It must be testable. So you can see some examples here of, alchemy versus chemistry, or phrenology versus neurology, magic versus physics, right? Well, you can't really test magic or alchemy or some of these things, right? Scientific theories have to be things that we can test. So that's key to our understanding here of these scientific theories, and the difference between a theory and sort of an idea, or sometimes the term theory is used differently in sort of common colloquial language than it is in science. In science, these are testable ideas that have withstood the test of time over many, many experiments. In terms of the data that we get in a scientific experiment, oftentimes we'll get something called qualitative data. And qualitative data, you can see the word quality in there, right? Well, the quality is not necessarily something that you can measure, but it's more descriptive data. It's maybe the color, the smell. So this guy who's digging in a sedimentary rock discovers this tooth. He's going to probably describe that tooth. That looks like a sharp tooth that might be found in a carnivore, something that eats meat as opposed to plants. So that type of descriptive data about that fossil is called qualitative data. And we'll oftentimes get qualitative data in our labs, and you'll describe colors or reactions and so forth using descriptive terms. The other type of data we try to get in biology labs is quantitative, and you can see the word quantity is in quantitative data, right? And so this is sort of hard numbers, right? Measured data. So we're going to use seconds, meters, liters, densities, things that we can actually graph, put in a data table, right? Compare to other sets of data, so numerical data. And so we'll be calculating in biology class quantitative data, hard numbers, and then also qualitative data, because qualitative data can also give us a little bit more information about a topic. So this is just a quick overview of the scientific method that we'll be using throughout this year in biology.